This presentation, we're going to be looking at the relationship between organisms and temperature. Temperature is one of the most important environmental factors and one of the most relevant in the face of today's rapidly changing climate. Why are they so important? Well, the fundamental importance of temperature to organisms, including you and I, is because the temperature is going to impact the rate of the chemical reactions in your body. So this is extremely important to the individual organism in order to maintain cellular respiration, to construct our proteins, for plants to do photosynthesis. And we're going to see throughout this chapter that all organisms are best adapted to a fairly narrow range of temperatures in which our chemical reactions can occur. And this is really defined by the enzymes that govern the rate of our chemistry. And these enzymes are proteins which work best in a narrow range of temperatures. So this is chapter 5 in the textbook Ecology, Concepts and Applications, written by Molles and Sher and published by McGraw Hill in 2019. I am Susan Glenn, and this is part of the lecture series for my ecology class. So the topics we're going to be talking about, we're going to give a little introduction with an example of an Arctic tundra plant, the driest plant. Then we're going to talk about the impact of microclimate, which is extremely important in your temperature regulation. We'll talk about evolutionary trade-offs, then look at how temperature affects the performance of an organism, look at how temperature can be regulated within the body, and then how organisms have adapted to survive extreme temperatures. Okay, if we look at this really pretty little yellow plant in the front of this Arctic tundra picture, that particular plant is Dryas integrifolia. And living in the Arctic tundra, obviously temperature is an important aspect of the Arctic environment. And if we're going to be talking about temperature, it probably makes sense to take a little bit of time to talk about what it is we're actually measuring. And temperature is a measure of the average kinetic energy, the energy of motion of molecules in a substance. So obviously if the molecules are moving faster, pushing each other around more, they have more energy, that gives you a higher temperature. So the kinetic energy is generally referred to as heat energy or simply as heat. And it is one of the most ecologically significant factors. So when you think about going up here in the Arctic tundra, maintaining a body temperature so that you can run your chemical reactions is going to be critical, and you need to maintain that for as long as possible. You can see that these little plants don't get to grow very big because there is a limited amount of energy available for a short growing season, and the sun is rather weak. It is not high above the horizon at any point in the Arctic. So they talk about, your textbook, they talk about a study in Ellesmere Island, which is very north in the Northwest Territories of Canada, 82 degrees north. Remember the North Pole is at 90 degrees north. And this is a researcher named Peter Kevin, went there in the summer, and what he had noticed was that as the sun moved across the sky during the day, the flowers tracked it like a satellite dish tracking a satellite. These flowers would track the sun in order to capture in sort of their parabolic shape, capturing that sunlight. And there would be close to 24 hours of sunlight every day, and these flowers would just keep their flower pointed towards it. That would heat them up and heat up their reproductive structures. So the flower is the reproductive organs of the plant. And it also attracted the pollinators, which are little insects that like to sit in those little warm flowers and go from flower to flower, and it would pollinate the flowers, and that way they could sexually reproduce. So here you can see the pretty little dryas flowers, and the light gets reflected and focused inward on the reproductive parts of the flower. So if the air temperature is 15 degrees Celsius, the temperature inside the flower is significantly higher at 25 degrees Celsius. And the insects can sit there and bask and warm up so that they can get the energy to fly to the next flower. So the topics we're going to be talking about, we're going to give a little introduction with an example of Arctic tundra plant, the dryas plant. Then we're going to talk about the impact of microclimate, which is extremely important in your temperature regulation. We'll talk about evolutionary trade-offs, then look at how temperature affects the performance of an organism, look at how temperature can be regulated within the body, and then how organisms have adapted to survive extreme temperatures. So microclimate, we've talked about microclimate a little bit. Microclimate is where the local landscape is going to affect the temperature. And when we were talking about the biomes, we were talking about the large macroclimate, which is the prevailing weather conditions in a region over a long period of time. Microclimate is the variation in the climate on the scale of a few kilometers or meters, or in some cases even centimeters, usually measured over short periods of time. So you understand microclimate if you've got standing out in the hot sun on the black parking lot, you're going to overheat. But if you move into the shade of a tree, move off that black parking lot onto the grass, you know it's going to be much cooler. So many organisms will move around, and it's going to influence their behavior, where they're going based on the particular microclimate. And for organisms, the microclimate may be more important than the macroclimate, that you can actually find a place that's nice and warm or nice and cool in order to spend the day there. And just your physical location or your behavior. And plants have behavior too, and we'll be talking about some of the plants as we go along. And obviously, when you're going up in altitude up to the top of a mountain, we know that the temperatures are generally lower at higher elevations. And so not only is temperature changing, but atmospheric pressure is changing. And that is the reason temperature is changing. So because as you go up, the atmospheric pressure decreases, and the air is rising up the side of the mountain, it'll expand then because the molecules are going to be further apart. That means the energy of motion required to sustain the movement of the air molecules is drawn from the surroundings, and it cools them. So that expansion of the air is pulling that heat out of the ground. And the second reason temperatures are generally reduced at higher elevation is that there's less atmosphere above the sites to trap and radiate heat back into the ground. So the clouds are not sort of pushing the heat back down. So if we look at the microclimate as we go up a mountainside, this is Mount Everest on this diagram. And as we move up Mount Everest, we can see that the air pressure is lower. So the red line goes from down on the ground, at an altitude of 0 kilometers, the air pressure is at 1,000. As we go up to the top of Mount Everest, the air pressures drop somewhere around 314. And if you went up higher in a balloon, you can see you could get even to lower and lower air pressure. As that air pressure decreases, then your temperature is going to decrease as well. So macroclimate would be the large-scale variation in weather. Microclimate is a small-scale variation in the weather. In this case, we're talking about temperature. And we've just seen that if we go up in altitude, we're going to find that the higher altitudes have lower temperatures because of the lower air pressure and the lack of atmosphere to re-radiate heat. Another aspect of a microclimate or another impact on microclimate is going to be aspect. And aspect is basically where you are in the landforms or the topography around you. Mountains and hillsides will shade certain parts of the land. Other parts of the land are going to be exposed to more direct sunlight. So it makes a difference whether you're standing on the sunny side of the house or you're standing on the shady side of the house. Where we live in the northern hemisphere, the shaded areas are always on the north-facing sides or the northern aspects of hills and mountains and buildings, which face away from the equator because the sun is always to our south here. Remember, the sun will be at the most directly overhead the Tropic of Cancer. It never is directly overhead this far north. On the other side of the planet, in the southern hemisphere, the southern aspect faces away from the equator, so that is the side that receives less direct sunlight. So because of the contrasting microclimates, the northern and southern sides of mountains can support very different types of vegetation. So microclimates and macroclimates are really referring to the scale of the weather variation, and we've been talking about the microclimate impacts of temperature and altitude, that the higher the altitudes, the lower the temperatures. We can also look at topographic features such as hills and mountains in terms of aspect, not just altitude. And that one side of the hill or mountain is going to have a shady microclimate and the other side is going to be exposed to more direct sunlight. And so that is going to influence the temperatures on the shady side of your house is going to be cooler and warmer on the sunny side of your house. So in the northern hemisphere where we live in the uh, in the U.S., the shaded areas are always on the north side of the buildings, the hills, the mountains, and the south sides or the southern aspects are facing the sun, and that's because we are north of the Tropic of Cancer, and the sun will never be directly overhead here. It'll never be to the north of us here. It's always going to be down to the south. It'll never be further north than the Tropic of Cancer, and the opposite occurs in the southern hemisphere. So the southern aspects in the southern hemisphere are facing away from the equator, so they receive less direct sunlight. So you can see how this microclimate, the temperature is also going to be affecting the energy, the amount of sunlight, and that's obviously going to affect photosynthesis. So because of the sunny and shady side of things, these aspects will offer quite different environments over a very short space. When you just walk around the edge of a tree or a forest or a building or a hill, you can immediately go from somewhere nice and cool to somewhere that's nice and warm. So here we can see an example looking at a north-facing slope and the versus the south-facing slope. And the south-facing slope in this particular diagram or this particular landscape is the one that has very little vegetation. The north-facing slope has a lot of trees on it, and the trees are on the cooler side of the slope, so it's not going to have the snow melt as quickly in the spring, and it's not going to dry out, so there's plenty of moisture for the trees, whereas the south-facing slope, so this side is going to be south-facing, and this side is north facing. So the sun is going to be shining directly on that south facing slope and drying it out. Okay, we see the same thing here. This is the woodlands, shrublands of the chaparral in Southern California, and we can see the same issue. We've got quite a strong contrast between a south-facing slope that is covered with grasses and it dries out and gets hot in the summertime, whereas the north-facing slope has a lot more shrub and tree cover on the left side, and that is going to stay a lot cooler throughout the day. And we can see that over a very short period, you're in completely different type of vegetation based on this change or difference in microclimate. There are other factors that are going to be influencing the temperature quite a bit, and that is whether there is any shade. The vegetation will shade the landscape, creating microclimate. Trees, shrubs, and even the fallen leaves, branches, and twigs can produce a little bit of shade in the desert. So every little bit of shade is going to count. And the desert landscapes, which have sort of a lot of wide open bare space as well as a little patchwork of vegetation, is going to have a patchwork of microclimate where things can be very, very different over a very short period, whether you are found down under these little shrubs or out in this open dirt landscape. So this is an example of looking at the microclimate where they looked in the desert to see what the temperatures were based on the location in relation to the vegetation. And we can see out here on the bare soil where you were out in the open from that last picture where there were not any shrubs, you've got a temperature out here of 48 degrees Celsius, unbearable temperature. Whereas even if you were a little insect and you were able to get down under the leaves that had fallen off of the shrub, you've dropped the temperature by 19 degrees. You're down to 28 or 29 degrees Celsius under the litter. So just crawling under the leaves makes a difference. If you get under a small shrub that's provide a little bit of shade, you can drop it a couple more degrees. And if you're getting under a larger shrub, obviously those bunches of large shrubs is going to give you the most protection from the heat, and your temperature is going to be much lower. The color of the ground can also significantly affect temperatures. You know that if you stand on a black fresh pavement in the summertime, it's going to feel really hot as opposed to standing on the grass or standing on lighter colored ground. And so this is the darker colors are going to absorb more light, and these are significant to the point that we will often characterize an area with the black volcanic sand versus the white sand, and the black volcanic sand is definitely going to hurt your feet more from the heat building up. Another example of something that's going to impact your temperature is whether you can get down into a shady burrow or whether you can hide under a boulder. Anything that's going to provide you a place underground or a place that's out of the sun is going to make a difference in your temperature. So this is some data that was taken from the Sevilleta National Wildlife Refuge in central New Mexico. This is an LTER site that's operated by the University of New Mexico. LTER stands for Long-Term Ecological Research Site. These are sites that are funded by the National Science Foundation where they collect ecological and environmental data every year in the same way. And there's a collection of these sites all over the country where they collect the same sort of information. And then scientists have a baseline of information and they can go and apply to do research projects in addition on these particular sites. And the Sevilleta site is found in the Chihuahuan Desert. And daily temperatures in the Chihuahuan Desert will fluctuate quite a bit. If you look at these air temperatures in this diagram, going from midnight to 6 a.m., we can see they can drop down to the low 20s in the morning, and then by mid-afternoon, 4 o'clock in the afternoon, they've gone up to over 35 degrees, which is unbearably hot. This is Celsius. And then late afternoon, evening starts to drop again. And so organisms being exposed to these temperatures are going to have to adjust their behavior depending on what it's like out that day. And then the soils, so if you burrow down into the soils, so let's say you're something like a kangaroo rat, you burrow down in the soils, you can see that the soil temperatures are actually much more stable throughout the day. And this was some research by some researchers at the LTER site so that you could actually see the impact of being underground. The picture across the top is a picture from the Sevilleta. That's a herd of pronghorn antelope, which is North America's fastest land animal and our only antelope in North America. Well, we know in the summer in New Jersey, when it starts to get unbearably hot, we try to go down to the shore so we can cool off with the cool ocean breezes. And we do know that water has an impact on our temperatures. So a little bit about water. And a little bit about its ability to absorb heat energy without changing temperature. That is a property uh, of uh, of substances that is known as the specific heat. So the capacity to absorb heat energy without changing your temperature is the specific heat. And the capacity for the specific heat for water is about 3,000 times higher than it would be for a certain volume of air. So it takes a lot more heat to heat up the water than it does to heat up the air. So if we look at water, 1 calorie of energy is defined as the amount of energy needed to heat up 1 cubic centimeter of water 1 degree Celsius. With the air, to heat up 1 cubic centimeter of air by 1 degree Celsius only takes 0.0003 calories of energy. So air is much easier to heat up. So air heats up during the day, cools off at night more quickly than the water does. So being close to water, the water can be absorbing a lot of the heat, and that way the water is keeping the surrounding area from heating up too much. The other thing water can do is that when water starts to evaporate, it is going to be absorbing a lot of heat. It's called the latent heat of vaporization. And this is the heat absorbed by the water as it evaporates. And that is a very important cooling mechanism. So when you get covered with water and then you come out of the ocean and stand in the breeze, the water evaporating off the surface of your skin is going to carry away heat from the surface of your skin. It also happens when you sweat. So when you sweat, you're putting water on the surface of your skin. That will evaporate away. And when it evaporates away, you're able to cool yourself off. So this is a great cooling mechanism for organisms. The problem is if you're sweating and cooling yourself by sweating and you're out in the desert, water is a limiting factor. So it's going to be hard to maintain the water you need if you're spending a lot of time cooling yourself with sweating. The last property of water that some people are not aware of, but it does give you more stability in aquatic environments, is the energy water gives to the environment as it freezes. And this is called the latent heat of fusion. And water gives up approximately 80 calories as 1 gram of water freezes. And so the energy of the motion of the water molecules decrease as they go from a liquid state into the crystalline state of the ice. So as a gram of pond water freezes, it gives off sufficient energy to heat 80 grams of water at 1 degree Celsius, and that prevents the water from continuing to cool. So that obviously is going to be an important source of energy in the wintertime as the surface of the water is freezing over. The organisms can appreciate some more heat coming that way. So the latent heat of fusion is another impact of water on temperature. So aquatic environments have a lot more thermal stability, especially the more water there is, the more thermal stability there is. But even if you have a small stream, you can find that the temperatures within the stream are going to fluctuate much less than the temperatures in the air around. And it's not just the water that's having a benefit, but there's also vegetation growing along the sides of the stream. And so we define this vegetation along stream sides as the name of riparian vegetation. So riparian areas are streamside areas, and they are protected by federal law because they're very important in maintaining the stream environment, which is also maintaining your water quality. So if we look at the way the temperature is being impacted along the side of the stream, we can see in this study the air, just the daily temperature changes in the air in this particular study was going from air temperatures around 4 degrees Celsius to air temperatures all the way up to 28 degrees Celsius. However, if you went and you were in these little reeds by the side of the stream, growing out of the stream surface, being in that shade and being closer to the water, you can see how drastically less variation. So it never got as cold and it never got as hot in those little reeds. The areas in the stream that are shallow, where the water is bubbling over the rocks, and you can see the clear water and the riffle habitat. Streams generally have riffle and pools and riffle and pool habitats as you go down the stream. In the riffle area, where it's a little more shallow, but you're still right in the water, we can find that the temperature variation is even less. And then you get down into these deep pools in the stream. You go down into the bottom of a deep pool, and you're going to find very little variation in temperature. So we're seeing the daily variation is somewhere closer to 4 or 5 degrees instead of this broad variation we saw of something around 24 degrees variation over the course of the day. So these deep pool environments are nice and cool and stable compared to the rest of the environments around it. So riparian vegetation influences the stream temperature by providing this shade. So this is the end of section 5.1 in chapter 5 of Temperature Relations. And you should be able to do the 3 concept review questions at the end of the chapter. For example, you should be able to explain how the microenvironment in a dryas flower could affect other species. And then what kind of advantage does this give to your dryas flower? So the topics we're going to be talking about, we're going to give a little introduction with an example of an Arctic tundra plant, the dryas plant. Then we're going to talk about the impact of microclimate, which is extremely important in your temperature regulation. We'll talk about evolutionary trade-offs, then look at how temperature affects the performance of an organism, look at how temperature can be regulated within the body, and then how organisms have adapted to survive extreme temperatures. The next section of the chapter talks about evolutionary trade-offs. And what we're going to be discussing here comes back whether we're talking about temperature, whether we're talking about water, reproduction, anything an organism does. Really adapting to one condition generally reduces your ability to adapt to other conditions. You cannot be good at absolutely everything. So we might imagine an organism that can live in any environment and thrive in all environment, but when we think about it in an evolutionary sense, that organism would have to have high fitness across all environmental conditions. So that could be some sort of superorganism. And as far as we know, there aren't any. So every organism has energetic limitations, and you can't be good at absolutely everything. All known organisms are adapted to a limited range of environmental conditions, and this is at least partially because of energy limitations. So this is leading to the concept of the principle of allocation. Organisms allocate limited energy to a certain function, which then reduces the amount of energy available for other functions. This trade-off in energy allocation will differ in different environments. So the functions might include growth, reproduction, and defense against predators. So an example might be if you're living in an environment that is lacking light and you're a plant, you might grow a bigger trunk so you can get up higher to get the light. Or if you're living in an environment where conditions change frequently, maybe there's a lot of flooding, you might put a lot of energy into reproduction, produce a lot of seeds so that you're able to make sure your offspring can get off into a different environment. If you're in an environment where you're getting eaten a lot, you might be a plant that has spines. If you're putting energy into growing spines, you might not have the energy to put into trunks or put into seeds. You can't put it into every function. So the researcher Levins, when talking about this, Richard Levins, he basically used a mathematical model to look at the evolutionary consequences of trade-offs, and published it in a book called Evolution in Changing Environment. And basically, he concluded that the evolutionary consequences of this trade-off and allocation results in populations having high fitness, high reproductive fitness, evolutionary fitness in one environment, but lowered fitness in another environment. And your textbook talks about testing, this is a hypothesis, testing the principle of allocation. And it describes the work that was done by Albert Bennett and Richard Lenski by looking at a microbial population. It is a nice thing about bacteria is they grow very quickly. They have a very short generation time. So if you want to study evolutionary changes in a population, then bacteria go through their population so rapidly, you can clearly be able to manipulate the environment and manipulate the temperature and see if there was a trade-off in their reproduction rate depending on the temperatures. So you can go through, with bacteria, you can go through hundreds of generations in a week. And this was looking at bacteria E. coli, which you probably are aware of E. coli, and they can live within the body at our body temperature. We're going to spend a little bit of time on this graph because it is a confusing graph, and the textbook does have a few of these scattered throughout, which is why I bother to explain these things to you. When you're looking at a graph like this, first of all, you kind of try to figure out what is the X and Y axis. Then they have usually some little notes that they've written on the graphs in order to explain things to you. So they put these little boxes on the graph to try to explain things. And then you read the caption underneath the figure. And then if it's still unclear, go back and reread that section of the chapter. So I'm going to reread this section of the chapter as we're talking about this experiment. So Bennett and Lenski's experiments looked at 4 different lineages of the bacteria E. coli. This lives in your body, so it usually lives at a temperature of 37 degrees. And they grew these 24 lineages at 37 degrees for 2,000 generations. They say a little bit later that the nice thing about bacteria populations is that they have such a short generation time. So a generation time for E. coli at 40 degrees is approximately 20 minutes. So they get a new generation every 20 minutes. They reproduce every 20 minutes. So you can do great evolutionary research with microbial populations. Then they took these populations and established 6 replicate populations by growing them at 4 different temperatures. So they would have time over 2,000 generations now to adjust to 4 different temperatures. They had them grown at 32 degrees Celsius, 37 degrees Celsius, which is their normal temperature, and 42 degrees Celsius. And then they had a 4th population that they alternated between the 32, the low, and the 42, the high. So they maintained these 24 populations. These are the 24 dots you see on this graph. They maintained these 24 populations for 2,000 generations, which gives them time to adapt to those 4 different temperature regimes. Then they used bacterial cells from each of their 24 populations to establish 24 new populations, which they all grew at 20 degrees Celsius for 2,000 generations. So theoretically, they're adapted to this low temperature in that process. Now, the original question, will adaptation to a low temperature of 20 degrees be accompanied by a loss of fitness at 40 degrees? Can you only be good at 20 degrees? You can't be good at 20 and 40, even though some of them had been grown at higher temperatures before. So they compared the fitness, and their measure of fitness was how quickly the population size doubled. So it's going to be their reproductive rate, which is really evolutionary fitness. They compared the fitness at the low temperature selected line with the fitness of the ancestral line at 20 degrees Celsius and 40 degrees Celsius, and to see whether the fitness when they were grown together was changing. So 2 major results stand out. First, the populations grown at 20 degrees Celsius had higher positive fitness at 20 degrees Celsius compared to their ancestors. In other words, they showed that the lines grown at 20 degrees Celsius for 2,000 generations had adapted to the lower generation, to that lower temperature. So if we look at what the x-axis is here, we have their fitness being grown at 20 degrees Celsius compared to their original ancestral lines. And so we can see if they are doing the same, there was no change in fitness. If they do worse, they grow slower, there would be reduced fitness. And if they are doing better than their ancestors, they have increased fitness, they're growing faster. And so in general, when they've been adapted to growing at 20 degrees compared to their original ancestors, they're growing faster. So everything except for one population, all the rest of the populations have improved fitness at 20 degrees. But then they looked at the lines that had adapted to 20 degrees. They had, in general, a lower fitness compared to their ancestors when grown at 40 degrees. So they took these populations, grew them at 40 degrees. So that's what the y-axis is. And so if they had no difference, if they're growing the same at 20 and 40 compared to their ancestors, they would be at 0 change in fitness. If they're doing better, they'd be up on the top half of the graph. And if they're doing worse, growing slower, they'd be at the bottom half of the graph. Excuse me. So we can see that for most of these populations, they're down below 0 on this y-axis. They're down in the bottom half of the graph, which means for most of these populations, they are showing a reduction. They're not as well adapted to growing at 40 degrees Celsius as they were at growing at 20 degrees Celsius. This result provided the first direct experimental evidence in support of Levin's principle of allocation. So the principle of allocation, remember, was saying that you could not be adapted to all conditions. If you were adapted, if you're doing well in one condition, that meant your fitness was lowered in another condition. So this principle in turn offers an explanation for the observation that most organisms perform best under a limited range of environmental conditions, including temperature. So this is section 5.2 in your textbook. It's a short section, but it's a very important section. It has a lot of important information. There's a couple of questions at the end of that section 5.2, and I would strongly recommend you being able to answer those concept review questions. So the topics we're going to be talking about, we're going to give a little introduction with an example of an Arctic tundra plant, the driest plant. Then we're going to talk about the impact of microclimate, which is extremely important in your temperature regulation. We'll talk about evolutionary trade-offs, then look at how temperature affects the performance of an organism, look at how temperature can be regulated within the body, and then how organisms have adapted to survive extreme temperatures. When we're talking about temperature, and we know now that most species perform best in a fairly narrow range of temperature, then when we look at the individual organisms and we can look at how the environment affects them, we can see that they are changing their behavior. They can be changing their ability to grow fast. They could affect how many offspring they produce, escape predators, catch prey. So all of these things put together is looking at the overall performance of an organism. Now, when we look at the performance at a molecular level, a biomolecular level, you'll remember from your general biology class that temperature is going to be affecting enzymes. Enzymes are proteins that help speed up chemical reactions. So enzymes usually work best in a limited range of temperatures. Most enzymes at low temperatures have a very predictable shape. The problem at a low temperature, your enzymes are shaped perfectly to help speed up your chemical reactions, but everything at low temperatures is moving kind of slowly. So remember, the molecules at low temperatures are moving more slowly, and so that will cause your chemical reactions at the temperature is low to be a little bit slow. So enzymes usually work better as they are warming up. But when you get to an excessively high temperature, remember your enzymes, your proteins will denature so that they will start to lose their proper shape. They will not be able to bind with the substrates, and they won't have the flexibility to do their particular functions. So there's an optimal range of temperatures for most enzymes. So if we look at, and we're going to be looking at this with a number of different examples, but if we look at how enzyme activity varies with temperature, remember on a graph, your x-axis, your horizontal axis, is going to be your independent variable. So the enzymes are not making the temperatures hotter or colder. The temperature is actually influencing the enzyme. So the enzyme activity is dependent on the temperature. So on the x-axis, I'm going to put temperature. And in this case, I'm not going to put specific numbers on here because that's going to vary with the organism. We're just going to basically indicate that you're going from a low to a high temperature, so you know what direction this is going. And then on the y-axis, I'm going to put some sort of a measure of my enzyme activity. So the enzyme activity can be measured in a number of different ways. For instance, if we were looking at photosynthesis, we might be measuring the enzyme activity based on the rate that carbon dioxide is taken into the plant for photosynthesis. When we look at the relationship then between temperature and enzyme activity, the ability of the enzyme to work, we know that if the temperatures are low, the enzyme really, everything's kind of moving sluggishly. When the temperatures start to increase, we can see that the enzyme activity is going to get higher and higher and higher. But then we start to reach a point where the enzyme will start to denature. And when we reach this point, we can see that the reaction rates will start to slow down. You'll reach a peak, and then it'll denature and fall apart and just not work anymore. So you get a curve, and we see these over and over again. And if you flip through your chapter, you will see these curves that show the enzymes will be working together or working better as temperatures increase. So this is increased movement rates of molecules with temperature, and this is the denaturing. So our enzyme starts to fall apart. These enzymes, remember, they're proteins. Proteins are made using the instructions on the DNA that your parents gave you. And so they must be important if that's what your parents gave you. That's all they gave you were the instructions to make proteins. And so these enzymes have to run all your chemical reactions in your body. Your body is such a low temperature for this amazing chemistry to take place that without these enzymes speeding things up at these low temperatures, you would not be able to complete any of your chemical reactions in time. You would not exist. So we need these enzymes to make everything else work within our bodies. So they then give you a really nice example in your textbook of looking at how these enzymes perform at different temperatures. And this was the work by Baldwin and Hochachka, looking at the enzyme acetylcholinesterase in rainbow trout. Now, let's say you have forgotten anything about acetylcholinesterase and acetylcholine, and you don't remember anything about enzymes. This is a good time to go back, look these things up. So we've assumed that you have some general biology and biochemistry knowledge about these things, and if you don't forget, go look these up. Make sure that you understand them. So acetylcholinesterase is an enzyme that's produced in the synapse between your neurons. So remember, in your nervous system, you will have the little spaces. So you have a chemical signal going down your nerves. Let me get a brighter color. You have chemical signals going down your nerves. And then when we're going to go to the next nerve, we have a little break between nerve cells called a synapse. And so we got an electrical signal sort of going down the nerve this way. It hits the synapse and it has to jump, and the electrical signal can't jump. So what you do is you produce a chemical signal, and so you're going to produce acetylcholine from this one nerve cell, will travel over to this other nerve cell, which will trigger it to continue the electrical signal down that nerve cell. Now, we can't have this acetylcholine just continue to sit here and making things just keep going when you don't need them going. So you need an enzyme that's going to come in and break down that acetylcholine. And when it breaks down that acetylcholine, then you can stop, you can relax. Okay, I don't have to keep this nerve tickling all the time. And so acetylcholinesterase is what is breaking down that acetylcholine in the synapse. So what did they find? So once again, when you get to something like this and there's a figure with it, and in this case it's going to be figure 5.8 in your textbook, and they describe your experiment and how they did it, read over these experiments, and I expect you to be able to do that. So Baldwin and Hoekstra in 1970, they published a paper about studying the influence of temperature on the activity of acetylcholinesterase, an enzyme produced at the synapse between neurons. This enzyme promotes the breakdown of the neurotransmitter acetylcholine to acetic acid and choline, so turns off the neurons, a process critical for proper neural function. The researchers found that the rainbow trout produced two forms of acetylcholinesterase. One form has the highest affinity for acetylcholine at 2 degrees, which is the winter temperature of the water, 2 degrees Celsius. However, the affinity for this enzyme for acetylcholine declines rapidly as the water warms up over the summertime. And there's a second form. They produce two different forms of the same enzyme. They're so clever, these fish. The second form of acetylcholinesterase shows highest affinity for acetylcholine at 17 degrees Celsius, which is in general the summer water temperature. The affinity of the second form of acetylcholine esterase doesn't work well at a lower temperature or a higher temperature. So they have two forms of the enzyme, and it works at two different temperatures. So how is this going to look? We have temperature as the independent variable, and we have how well the acetylcholine works. So your enzyme substrate activity, so how well the enzyme will actually bond with the acetylcholine. So, and we're going from low to high temperatures here, and low to high activity there. It's amazing how many times on an exam people will draw a graph and they won't label low and high, and they'll do it backwards and the graph looks backwards. Sometimes they will put the dependent variable on the x-axis instead of the independent variable. So this is the independent variable. This is the dependent. It's very hard to determine, interpret it if it's mixed up. So, and actually in this case, I can put some numbers on here. I can put 0 degrees, water's frozen, obviously nothing's going to be working then. And then I could put things at 10 degrees Celsius, so this will be Celsius, 20 degrees Celsius, and 30 degrees Celsius. So I know there's two forms of this enzyme. So I'm going to draw two lines on here, one for the first form, one for the second form. The first form, as it said in the description, has a very high affinity, works best at 2 degrees Celsius. So at 2 degrees Celsius is where it's going to peak. So I can take this first form, and I'll call it the cold water form, is that when it just gets above freezing, it's going to start working better and better as it gets warmer and warmer. And then, actually, I'll make this one, this is the cold one, so I'll make this one blue. And then when we get to 2 degrees Celsius, this is where it's at its peak, and then it'll start to degrade as it gets warmer, and then it drops off as it gets warmer and warmer, as the protein denatures. As that one enzyme is starting to fail, then the second version of this enzyme is going to start to kick in and start to improve, going faster and faster. And that one peaks at about 17 degrees Celsius, and then it will denature. So we have two different forms. We have the form that is peaking at 2 degrees Celsius, and we have the second form that is peaking at 17 degrees Celsius. So we have two curves for this. And that's the picture from your book, which is much better than the one that I drew. But you should be able to reproduce something like this on an exam question. So we want to move on to talk about plants, but they do give you another example of animals in your textbook with a different way of measuring metabolic activity, and that was the eastern fence lizard, which we do have in New Jersey. So make sure you read that over, and they are looking at the performance of the animal in terms of its rate of metabolism of food. I'm going to move on now to talk about how plants are also influenced by temperature through the process of photosynthesis. So this is obviously a chance for you to review what photosynthesis is. And this example is looking at two plants that have evolved to grow in very different environments. We have a moss growing in the boreal forest that is growing on the forest floor, so it's a very cool area that it is growing up north and in the shade, versus a desert shrub, which is Atriplex lentiformis, and they talk about this desert shrub various times in the textbook, so it might be important to kind of get interested in this or familiar with this shrub. And once again, with these plants, we can see they're adapted to very different environments. So on the x-axis, we have the temperature, which is in degrees Celsius. That's the independent variable, going from very low temperatures to high temperatures. And then on the y-axis, we have the photosynthetic rate. And instead of giving you the actual rate, maybe of CO2 intake or something like that, it's doing it as a percentage of their maximum, because obviously this desert shrub that is out here in the sunlight in the middle of the desert has a much higher photosynthetic rate than the moss, which is sitting in the shade in the forest. So they want you to be able to compare these the shapes of these two graphs and where their peaks are. And we can see the boreal moss shows exactly the same patterns we saw when we were talking about the enzymes in the fish, that as the temperature is increasing, the photosynthetic rate is going up. Its peaks as it starts to denature, so you find the peak at 15 degrees Celsius, and then it'll slowly start to break the enzyme down till you couldn't grow that moss at a very high temperature. And even if you took that moss and decided you were going to take good care of it and grow it in your house is at 22 degrees Celsius, and it's not going to do as well in your house as it was doing out in the forest floor. This is in contrast to this desert shrub. The desert shrub really could not grow at 15 degrees Celsius. Its enzymes are not really going to kick in until it gets much warmer than that. And then they peak at 44 degrees Celsius, which is incredibly warm, and then they will rapidly denature after that. So they're not going to be able to function at a much higher temperature. So obviously these are genetically adapted to thrive in the environment. They're very different environments, the boreal forest and the desert biomes. If you're asked to draw the photosynthetic rate of a moss from the boreal forest and a shrub from the desert in relation to temperature, you know to put the temperature across the x-axis. That is the independent variable. And in this case, you could probably even put some numbers on it. You could probably put 0 degrees Celsius and 50 degrees Celsius, or 60 degrees Celsius across there. Or at the very least, you could put low to high. And then on the x-axis or the y-axis, we have the dependent variable, which in this case was the percent of photosynthetic activity. So percentage goes from 0 to 100. You have one line for the moss. You have one line for the shrub. Actually, let me erase those. One line for the moss. They have to go to 100%, right? And that 100% for the moss was around 15 degrees Celsius. And then the shrub, we know, started around there. And the shrub peaked at 44 degrees Celsius. So you even have a couple of little numbers you could put on this. And there you go. Now, when we've been talking about adaptation to different environmental conditions and temperatures, we've been talking about genetic adaptation of populations. However, we can also talk about adaptation of the individual, and that is acclimation, where you have a short-term physiological adjustment, and that allows you to respond to the temperature. So this study is the same plant we were just talking about, that desert shrub, the Atriplex lentiformis. And this was done to see if it could acclimate, if it grew in different temperatures, if it could change its photosynthesis. And this was a work done by Robert Percy, and he clearly demonstrated the effect of acclimation on photosynthesis. So he took a population of the desert shrub that was growing in the Death Valley in California, a very hot environment, and he took cuttings of them. So he had cloned plants, propagated plants from the cuttings so that they would be genetically identical clones. And the clones from the Death Valley plants then were grown under two different temperatures, one in a hot set of conditions at 43 degrees Celsius during the daytime and 30 degrees at night, and the other was under cool conditions at 23 degrees during the day and 18 at night. And then he measured the photosynthetic rate of the two plants. And as you can see, the plants that were grown at the daytime temperatures at 43 degrees had a much higher peak in their rate of photosynthesis. So they were peaking at around their daytime temperature over 40 degrees Celsius, and the clones of the shrubs that he grew at the cooler temperatures were peaking. It's still higher than the temperature that they were growing at, but it was much higher, much lower. So it's about a 10 degree Celsius difference in the maximum rate of photosynthesis between the two different populations. So this is a physiological adjustment, and so we know that individuals can acclimate to different temperatures and adjust their physiological responses. So the enzymes that were working at one temperature are different than the enzymes that are working at different temperatures. So this means that plants and animals can acclimate to the changes in temperatures with the seasons. We saw this with the enzymes of the acetylcholinesterase and the salmon, and now we see it with the photosynthesis and these. And we know in this case, how do we know that these were not genetic differences? Remember, he took clones. He took genetically identical individuals to do this study. Now, we have been talking about photosynthesis. I wanted to take a moment to review what photosynthesis was in case you've forgotten. So remember that the plants have to take in carbon dioxide because they're going to, in photosynthesis, they're making sugar. So we need carbon for sugar, so they're taking carbon dioxide. Where's that carbon dioxide coming from? Well, that's sitting out in the air. How does it get into the plants? Into the leaves. The plants don't have lungs, so into the leaves of the plants. So the leaves of the plants have little holes on most of them on the underside, called stomata or stomata, and that allows the carbon dioxide to come in. They will also need water. The water is in the soil. So that's sitting down there in the soil, so it's going to come into the roots of the plant. So we're going to have the roots down here in the soil. It's going to come into the roots, and then it's going to have to go up the stem. And when it gets to the top, it's going to go onto the leaves where it can mix with the carbon dioxide, and we can go through a whole series of chemical reactions in the presence of sunlight. So remember, we are also going to need the sunshine here. So we have the sunlight, which is a source of energy. And through the process of the light-dependent reactions, that sun is going to split this water. So it's going to break this water up. And when it does that, it's going to take the electron from the hydrogen and it's going to heat that up. And we're going to use that electron to generate some ATP. And the plant is going to get rid of the oxygen. So the oxygen is a byproduct that is released. And that's released back out into the air through those very same stomata in the leaf. So it gets rid of the oxygen. We still have the hydrogen. We're going to make sugar in the light independent reactions. So remember, the sugar is the glucose. It's C6H12O6. So we're going to produce some glucose there, and that glucose can go and be, so we use the ATP, remember, from the sun and the electron and the hydrogen, so that glucose has been glued together with all of that energy. That energy can be used in cellular respiration then, so we can use that energy to for the plant to do its chemical reactions, to grow, and we can also store it, so we can put that glucose. So remember, this is glucose, so we can use it, or we could store it, and the plant will make long chains of glucose, and which is starch, which they could take down and put into their roots and use later. So it can store as starch, which is chains of glucose. In order to balance this equation, though, you're going to have to take in 6 carbon dioxides, and we're going to have to take in 12 waters in order to make all of this work. And you're going to get 1 molecule of glucose out of that, but you're going to have 6 molecules of oxygen gas that you release. And then you're also going to have to take, remember we were ripping apart this water and we've got some hydrogen left over from that. And so we're going to take that hydrogen and we're going to put that in the form of water. So that's an H2O. And so that water is going, there's going to be 6 of those produced as well. 6 waters. And that water is also going to go out into the air through the stomata in the leaves. This release of water into the air is significant. This is the evaporation of water into the air, so this is going to be water vapor. And this process of evaporating water from the leaves of the plant is called transpiration. And if we're talking about the evaporation of water, whether that's sweat or whether that's from the leaves of the plant, that's going to cool the plant. That's going to affect your temperature as well. So that is going to have a kind of a cooling effect. Okay, so we do know that extreme temperatures are going to affect the enzymes in photosynthesis. So here's the equation I just wrote for you. Extreme temperatures will usually reduce the rate of photosynthesis, and this is an issue for global climate change because we're finding the tropical rainforests are getting so hot, the trees are not able to complete photosynthesis. The enzymes are breaking down. And we know that the plants have different optimal temperatures. So we've talked about animals, we've talked about plants. Let's go back to looking at microbes. And there are microbes that seem to live in all temperatures around the world. You can find them in cold waters of Antarctica, the boiling waters in the Yellowstone hot springs. However, these extreme environments have specific microbes that live there. There's no microbe that can live in all the environments. Marita studied the effect of temperature on the population growth on the bacteria that loves living in cold water, cold-loving or psychrophilic marine bacteria around Antarctica. And he took these bacteria and grew them in a temperature gradient incubator for 80 hours. And during that time, he ranged the temperature from -2 Celsius to over 9 degrees Celsius. And he found that these bacteria grew their fastest at 4 degrees Celsius. So even though if you got the water warmer, it wasn't going to make them any happier. He even found that they were able to grow in super cold water. Remember, salt water is going to have a lower freezing point. He could even find that he could grow it in super cold water at -2 degrees Celsius And even some of the bacteria were growing at minus 5.5 degrees Celsius. On the other hand, some microbes can live at very high temperatures, and they have found microbes living in all of the hot springs that have ever been studied. So these ones that love the warm are called thermophilic, and they can grow at temperatures above 40 degrees Celsius is how they're defined. And so many of the heat-loving microbes can only live in those temperatures, so that they are hyperthermophiles, which means they have temperature optima over 80 degrees Celsius. And some of them can even grow as high as 110 degrees Celsius. So the most intensive studies of these have been done at Yellowstone National Park, and they were able to see that even some of these guys were able to get their energy by oxidizing sulfur as opposed to using the energy from the sun. And it's interesting because since we know that they could do their chemical reactions at such a high temperature, it meant that they had enzymes that were not degrading at these extremely high temperatures. So we actually use those enzymes when we're doing PCR, when we're doing DNA replication, in order to make sure that we can separate the strands of the DNA. We heat them up, and we don't want the enzymes to denature, but we took the enzymes from these bacteria and have been able to use that. And that's why PCR was possible. And we couldn't do PCR until we discovered some organisms that could work at these extremely high temperatures. So you can heat your DNA up to 90 degrees Celsius, separate the DNA strands, and the enzymes will still stay intact. And then you can cool it and do the replication on each side of the PCR, or each side of the DNA strands. So this is looking at the graphs of the growth of these bacteria. And so we have the bacteria that were growing in Antarctica, and we're looking at on the x-axis, once again, we have the independent variable being temperature, and on the y-axis we have the rate of population growth being our measure. And we can see we have the peak of the population growth in these Antarctic bacteria at 4 degrees Celsius. The second graph here is looking at these bacteria from the hot springs in Yellowstone. Excuse me. And so we can see once again, we have temperature across the x-axis, which is it's our independent variable. And here the rate of sulfur oxidation. So this is the rate that they are using the sulfur for their energy source is being measured. And we find that their peak is around 60 to 63 degrees Celsius. And the hot spring itself was at around 59 degrees. So they're really operating right at their peak amount of peak photosynthetic rate at the hot spring temperature. So that's the end of section 5.3 in your textbook, and you should be able to answer the questions at the end of the concept review questions there. First one was asking about signs of thermal stress in fish swimming on their sides and swimming in spirals. Explain, using what you know about temperature and acetylcholinesterase, explain what's going on. So think about what acetylcholine does, what acetylcholinesterase does, what happens when that denatures and stops working. So you should be able to answer that question. And then the second one is, how can we be sure that two distinctive responses to temperatures shown by Atriplex lentiformis were due to acclimation as a result of the genetic differences? And it's So the topics we're going to be talking about, we're going to give a little introduction with an example of an Arctic tundra plant, the dryas plant. Then we're going to talk about the impact of microclimate, which is extremely important in your temperature regulation. We'll talk about evolutionary trade-offs. then look at how temperature affects the performance of an organism, look at how temperature can be regulated within the body, and then how organisms have adapted to survive extreme temperatures. Because our body temperatures are extremely important for our physiology, it seems like we don't just let it do whatever it wants. Organisms have many different approaches and many different ways that have evolved that allow them to regulate their body temperature. Whether they're cold-blooded or warm-blooded, the organisms still have to make sure their enzymes work. I put the picture of the zebra here because one of the theories about its stripes are that the dark stripes will heat up the air, which will rise and cause movement of the cooler air from the white stripes. It causes air movement across the surface, which will help the animal regulate its temperature. It's like making a little breeze on its skin and helping evaporate water off of its skin at the same time. So your textbook takes these factors that will allow the organism to balance its heat gain with its heat loss by using this equation that sort of helps put it all in one place. And so the equation, I know it looks worse than it really is, but here we're looking at H sub S. So we're looking at the total heat stored. So the S is the storage of the heat in the body of the organism. And that heat that's in the body, part of it is gained from metabolic heat. And metabolic heat is the energy that releases when you're doing cellular respiration. So you think about when you are running around and you're burning up a lot of sugar, you get hot, you heat up. That's the heat from your metabolic rate. So you gain heat from metabolism. Then we have the CD is for conduction. Conduction is the transfer of heat between an object and direct physical contact. So when you sit on a cold bench, a stone bench on a cold day, you're going to lose heat from your body into the stone bench. When my cat sits on my lap, she'll absorb heat on the cold day into her body through conduction. So you can gain heat or lose it. And that's why in the equation itself, it has a plus and a minus on it. Because you could be gaining or losing heat. Same thing with convection. Convection is the same process, but it's the process of heat flow between a solid body and a moving fluid. That can be the air, so that the wind carrying your heat away, or that could be in the water. You get in the water and the water will take your heat away. And so the direction of flow for heat for both conduction and convection is always from the warmer region to the colder one. So heat is going from a warmer spot to a colder spot, whether that is in or out of the organism depends on the relative differences. Then we have heat gained and lost through electromagnetic radiation. So they have an R for radiation. So this is often called simply radiation. All objects give off electromagnetic radiation, but the obvious source in our environment is the sun. And we don't really see this because well, really, most of the sunlight falls outside of what we can see. We can see the visible light, but there's an infrared, a heat portion of the spectrum. And the electromagnetic radiation emitted by most objects in our environment is in infrared light as well. So if you wore infrared night goggles, you could see the heat being given off of the warm bodies. When you stand in front of a fire, you're feeling the infrared heat from the fire. And so when you're standing outside on a clear, cold night with no wind, the chilling effect is your body giving off heat to the surroundings, and which eventually goes off into the night sky. And then the H sub E, this has only got a negative sign. So the electromagnetic heat, you can absorb heat or you can give off heat. The evaporation is the heat that we're losing from cooling our body with sweat, and it's the heat that the plants will lose through transpiration. So this is a figure from your textbook looking at a plant and how heat exchange is occurring between the organism and the environment. This is exactly the same as that equation, except in this case it's much more visual. This is much better for the visual learner. And so we have a plant here. A plant, it doesn't maintain a high body temperature. A plant is a type of organism known as a poikilotherm. A poikilotherm's body temperature varies with the temperature of the air around it. So you spell poikilotherm is Poikilotherm, poikilotherm. And so poikilotherm's body temperature varies with the environmental temperature. And so it has to use some sort of external source of heat. And so we're looking at this particular plant, and when we're really, let's say this is a sunflower living out in the desert, we can see the sun beating down on it is going to be bringing heat into the plant, so that's going to raise its temperature. But then remember the plant is also going through metabolism, metabolic activity. It'll give off a little bit of heat, but not a lot. So this desert plant is not giving off a lot of heat. It's going to give off more heat with evaporation of water through transpiration. And that's going to be pretty big. It's going through photosynthesis. Its stomata are open to let carbon dioxide in. So remember, CO2 goes in here, and water vapor and oxygen come out of the leaves. That water vapor is going to be evaporating transpiration, so that's H sub E. We have the wind blowing, so you can see this is a fairly open plant where there's lots of room for the wind, and they show the wind coming through the plant here and taking some of the heat away. But you can also imagine if there was hot air, so maybe it's heating up from the ground, and that hot air is coming up, and that's going to help heat up the plant as well. And then you have heat loss or gain depending on how hot the soil is. So you can have conduction down here. These desert plants tend not to put any leaves down on the soil surface because those would heat up as the soil heats up. So they tend to avoid that and keep their leaves away from the surface so that their heat of gain from the soil can be minimized as much as possible. So poikilotherms, the body temperature varies directly with the environmental temperature. And so they're going to have to regulate their body temperature by manipulating the evaporation and the radiation portions. The animals that mainly rely on external sources of energy for regulating their body temperatures are called ectotherms. And organisms that rely on our own metabolic heat reserves, our metabolic heat energy is called an endotherm. So we heat up, you increase you burn more sugar in order to heat up. And then we have endothermic animals. Some endothermic animals, including some fish and insects, can heat up critical organs, but not the whole body. Other endotherms are very good at maintaining a constant body temperature, and we are called homeotherms. And homeotherms include birds, mammals, and there's one deep sea fish, the opa. Is a homeotherm, so that you're able to keep a constant body temperature. And so that way all your enzymes will work really nicely within your constant internal body temperature. But that means you have to regulate it and keep it where you want it to be. So the desert plant that I was just talking about, desert plants have to reduce heat storage. So what we're looking at with the desert plants that we've got the stored heat uh, equals the heat from conduction, so the hot air around them, minus the heat that they could lose from convection from the wind, and they can try to reduce how much radiative heat that they are going to be bringing in. They can't really rely on evaporation because there's a shortage of water. So these are the factors that they have to deal with. So you want to reduce the heat with conduction, not put your leaves touching the ground. You want to increase conductive cooling. Make sure you let that wind get around you and cool you off. And you're going to have to try to reduce how much you're exposed to the sun. So here's a desert plant. In order to reduce conduction, you don't have the leaves are not touching the ground down at the bottom of the plant. In order to let the wind through, you have very sparse leaves so the wind can get in and around you and cool you off. And then what they will do is also orient their leaves so that they are not facing flat on into the sun, so they're not getting the whole brute force of the sun hitting the whole leaf blade, but they will turn the leaf sideways and it'll just be the edge of the leaf that heats up so that they are going to reduce how much of the leaf is exposed, and they can orient the leaves and move them throughout the day. So we saw with that dryas, it was moving its flower to capture the heat from the sun. Here we're moving the leaves to try to minimize the heat from the sun. When we look at Arctic and alpine plants, then they're going to have to try to stay warm. They want to increase their radiative heating, like that dryas flower. They want to decrease their convective cooling, so they don't want to have a lot of space for the wind to blow. They want to keep everything all tucked in like a big fat penguin, but these are plants. The tropical alpine plants are usually rosette plants, and what they do is in order to prevent their stems from freezing, they try to keep the dead leaves. So if we have the ground here and we have the plants growing up out of the ground with their leaves, what they do is keep a mass of all the leaves. So when their leaves die, they don't drop them. They will just hang on to them, and there will be dead leaves down here that'll help insulate the stem and keep the stem from the plant from freezing. They also have thick leaves with little hairs all over the surface. So they have very hairy, they're not hairs like our hair, but little projections, hair-like projections all over the leaves. So they're all fuzzy. And that increases the leaf temperature, keeps it nice and warm, holds in their metabolic heat. So here is a typical Arctic tundra plant, and these are known as cushion plants. You also get it in the alpine tundra, and so there's one of the things they will do. They'll have their leaves are very dark green, so remember that's going to absorb more light if it's dark, and that way they reduce how much light is being reflected. So they're trying to take in as much light as possible, so we can maximize the light coming down to the plant and into the plant to heat it up. They make this cushion shape so that when the wind does blow, it blows over them and doesn't blow. It's not going to blow through them. So since it's not blowing through them, you're not going to be losing heat from that. And then if the soil around them heats up with the sun, it's nice and close to the ground, so it can heat up based on the heat around it, so that it can increase the heat gain. So this graph is looking at an Arctic cushion plant compared with an Arctic willow. Now the Arctic willow is a more open plant. The cushion plant is the one we just saw in that diagram. And when we look at the or over the course of the day. So we're looking on the x-axis. We've got the time in the day in hours from 9 a.m. to 5 p.m., so morning to afternoon. And we can see the air temperature is the bottom line. So we've got the air temperature going up over the course of the day. Then we have the temperature of the willow, and the willow has a more open structure. It allows the wind to come through, and it basically is tracking the air temperature. It's not much warmer than the air temperature. However, the cushion plant is able to retain more heat, and so it's the top line here, and we see the cushion plant does much better and is much warmer than the air temperature. And so this seems to be a strategy that works for it. Now, ectothermic animals, remember uh, things like fish, amphibians, reptiles, and vertebrates, they use external sources of energy to regulate their body temperatures. And they're going to use similar ideas or systems as the plants do, but the animals obviously have more options because they can move around. The plants are kind of stuck in one spot, but we can see the behavior of the animal is definitely going to be an important factor here. This is one of the reasons animals do the things they do. So earlier in the chapter, they introduced the eastern fence lizard, and we're looking at the eastern fence lizard again. Here is an ectotherm that uses its behavior in order to regulate its body temperature. And if it's cold, it will go and bask out in the sun, and then if it's warm out, it'll go down and it's going to get in the shady spots. Its energy intake from earlier in the chapter is maximized at 33 degrees Celsius. And so its preferred temperature is close to 33 degrees Celsius. And that's the way it can get the most energy out of its food. Its enzymes want to work the best. This is what this really cute guy looks like. He lives on fences and stone fences, wood piles. You'll see him basking on the top in the spring, where he's going to make sure that his temperature gets up high enough. We find these in New Jersey, but they also they looked at it in this particular study. They looked at a population in New Jersey and a population in South Carolina. So there was a temperature gradient from 26 degrees in New Jersey to 38 degrees in North Carolina. And so no matter where they grew, where they were living, where the population was from, they both had their metabolic energy was maximized at 33 degrees. So the light blue, we've got the population from New Jersey. If they were living at 30 degrees, they were not metabolizing as much energy as at 33 degrees, and then less energy at 36 degrees. And the same thing with the South Carolina population, less energy metabolized at 30 degrees, 33 degrees, 36 degrees, less energy. So they both had their preferred temperature is going to be 33 degrees Celsius. However, remember, they live in different places with different temperatures. It's cooler in New Jersey than in South Carolina. They have exactly the same preferred temperature in both places, 33 degrees. And when you actually measure their body temperature out in the field where they're living, they have exactly the same body temperature. And that is because they're going to change their behavior. So in New Jersey, they're going to spend more time sitting up basking in the sun than they do in South Carolina, so that they're able to raise their temperature in New Jersey when they have to, and they'll go down in the shade in South Carolina when they have to cool off their temperature. So they end up with exactly the same temperature. So the two populations both regulate their body temperatures to match the temperature of their maximum metabolic energy intake. This is a really interesting example of grasshoppers. Grasshoppers also like to bask in the sun, which can raise their body temperature. And they actually found that the ones that were reared at low temperatures tended to be darker in color. And this darker color allowed them to absorb more heat. So what these grasshoppers would do is that they would go out in the sunlight in the morning, and these were grasshoppers that were living in the mountains of Arizona, and they would go out in the morning in the sun, and they could heat up their body temperature quite quickly that way to their optimal temperature. And they took them into the lab to see uh, what they were doing and what temperatures they were reaching, and some of them would even go above their optimal temperature, sort of overheat in the sun. They raised some grasshoppers in without access to light, and they raised some with access to light. And the ones without access to light actually were a darker color, so they would be able to absorb more heat than the ones that actually were going out into the light. And if they're a lighter color, they're reflecting more light, so they try not to overheat. But they were trying to figure out why they were actually basking to such a high temperature, and they realize that those high temperatures may have to do with raising your body temperature like you do when you get an infection. It kills a fungus that affects grasshoppers. So just like you would raise your body temperature to kill a bacterial or viral infection, they are raising their body temperatures so that they can kill this fungus infection, which is controlling one of their serious diseases or pathogens that they're affected by. So when we look at the data from the lab, the grasshoppers that had access to light were raising their temperatures very high. So this is over the time of the experiment on the x-axis, and we're looking at the temperature of their bodies or of the air, and we can see the air temperature was fairly constant during this time, which is the blue line across here. We have the temperature of the grasshoppers that were confined to the shade. They didn't have access to the light, so that tracks pretty closely with the air temperature across here. And then the grasshoppers that did have access to light were about 10 degrees warmer than the air temperature. Their body temperatures were much higher and stayed higher through the course of the time. Recommend taking a break here. We're going to talk about endothermic animals and how they regulate their temperature. It was usually with adjusting our metabolic rate. We do more metabolic activity to heat up, and then we do less to cool off. But these are a couple of really cool examples of kangaroos in Australia living in hot deserts and the mechanisms they use for cooling themselves. So take some time, watch these two YouTube videos, one on the eastern gray kangaroo and one on the red kangaroo. We'll be talking about these kangaroo species later in a later chapter, and take a moment and stop the video and watch these two examples, which are very interesting ways of the kangaroos cooling themselves. Now that you're back from watching those kangaroos keep themselves really cool, we're going to talk about temperature regulation by endothermic animals. And these endotherms are obviously going to rely a great more on the metabolic heat. And so there was a study by Scholander et al. In 1950 where they looked at thermoregulation in mammal species by monitoring their metabolic rate when they put them at different temperatures. And they found a range of environmental temperatures over which you didn't change your metabolic rate, so that the homeotherm had a range of temperatures where it really didn't have to do anything different metabolically, and that is called the thermal neutral zone. And that differs with different species. It can even differ with different populations of a species. If the temperature falls below or rises above the thermal neutral zone, then the homeotherm's metabolic rate will rapidly increase to 2 or 3 times what its resting metabolism is. And Well, basically at low temperatures, if this was happening to us, if the temperature dropped below our thermal neutral zone, we would start shivering, which causes our muscles to contract. We also release hormones that increase our metabolic rate, and we start metabolizing our fats. And at high temperature, what we will do is increase our heart rate, and that will push blood to the skin, and that can dump heat from the skin radiating out into the surrounding environment. So you turn red and you start to dump heat outside of yourself. And some organisms they talk about in your textbook will actually have special structures to increase how much they are dumping into the environment. And evaporative cooling from sweating, that can certainly unload a lot of heat. And humans will do that, horses will do that, camels will sweat to reduce their heat. Some homeotherms do not sweat, but they can cool by other means. So the dogs, birds, marsupials, they can pant. You saw the kangaroos where they will lick themselves so that the evaporating water will cool them off as well. And so there's a number of different techniques homeotherms will use to keep themselves cool. I find this a fascinating graph. What we have across the x-axis are the environmental temperatures of different mammals. And the most mammals have a normal body temperature around 35 to 40 degrees Celsius. So, and that falls within the range of the thermal neutral zone for the mammals. They all go into this range where we have our normal body temperatures. However, if we look at the tropical species, they don't go far out of that range. The tropical species have a fairly narrow thermal neutral zone, and if you get lower temperatures than that thermal neutral zone or higher temperatures than that thermal neutral zone, they have to increase their metabolic rate, change what they're doing physiologically so that they are still able to survive in those temperatures. And that includes us. We are a tropical species. We evolved in a tropical environment. However, take a look at these Arctic species. They're amazing because they have this huge breadth of a thermal neutral zone. For instance, the Arctic fox can tolerate environmental temperatures down to minus 30 Celsius without showing any increase in metabolic rate. Take a look at this Eskimo dog that can lie out there in the snow and it's well below freezing and it is quite happy and it is not having to adjust its metabolic rate at all. But then it can come here and live in your house and be just as happy. So I think this is a really interesting study where they were looking at these different species, thermal neutral zones, and comparing them between tropic and Arctic species. So the rest of this section of the chapter, it covers a number of other endothermic organisms that are kind of interesting. It talks about endothermic aquatic animals and the idea of using a heat exchange of your blood vessels to keep them from cooling off the body and the water. We also get these heat exchange devices in the extremities of birds, like you can see the feet of a duck standing on the ice, and as the blood goes down to the feet, it dumps the heat into the parallel arteries. And that way, as it moves back, it can take the cold blood and heat it up as it's going from the blood that's going back out to the feet from the heart. So they talk about these various types of fish and aquatic fish and animals that are mammals that are homeotherms, endothermic, and also some interesting endothermic insects and some of the work of Bernd Heinrich. So you can read about that. I'm going to skip on ahead to something that you wouldn't think was a thermogenic organism. A plant is a plant that does grow in New Jersey. Most plants are poikilothermic ectotherms, so their body temperature will vary with the air temperature, but plants in the family ericaceae use metabolic heat to actually heat up their flowers in the early spring to attract pollinators. So we're going to talk about the skunk cabbage, which is a perennial plant that has a big thick taproot, and it stores the glucose in long chains of starch in the root. And then in the spring, when things start to early spring, start to warm up. Sometimes you get those warm spring days, and what it'll do is it'll translocate the starch up to the flower. An inflorescence is a flower cluster, a flower structure, and it'll start to go through cellular respiration. So it goes metabolizes this starch into heat. That will melt the snow above the flower. And so you get this little purplish flower up here in the snow. And it gives off a skunky smell, a nasty smell that attracts flies. And the flies are the pollinators. So on a warm spring day, some of the flies are going to pop out and they'll be flying around. And the only place they're attracted to then is going to be these stinky flowers where they're getting tricked thinking they're getting some rotting meat, and they're going to go flower to flower, and there's no other pollinators out there, there's nobody competing for it, and they're able to take advantage of this metabolic they were generating. So here's a picture from your book of showing you that the equations that we've been talking about has it generating quite a bit of metabolic heat in the flower, which is going to melt the snow, and it's going to just stick there up above the surface. So the air temperature can be minus 15 degrees Celsius, and this little guy's poking up its flower out of the snow and has a nice flower temperature of 20 degrees Celsius. So quite a difference of 45 or 35 degrees Celsius difference in temperature. These skunk cabbages, here's a bear sitting in the skunk cabbages. They're kind of the first things to grow in the spring. Excellent form of vitamin C, so for animals and people. So how does the metabolic rate of skunk cabbage flowers change with the air temperature? So if we're going to do one of these graphs again, my pen has lost its charge, so I'm using the mouse. So we have the temperature across the x-axis. We have the metabolic rate on the y-axis. We're going to go from a temperature of 0 Celsius, the freezing point, to a temperature of 20 Celsius, which is about your room temperature today. And remember when we were looking at metabolic rates, we had the rates would increase with temperature, and then the proteins would denature. Well, actually, with these skunk cabbage enzymes, the proteins start denaturing as soon as the temperatures start to increase. So we find that with these skunk cabbages that the reaction rate is highest right when the snow's melting, and then it drops off from that point on. Really, the part of the line where it was going up with temperature would be back over here. We'd have it a line going like there where it would increase, but because the temperatures are below freezing, they can't do anything. So it can't even start functioning until the temperature is above freezing. And here it is in the textbook, much better picture than I just drew with my mouse. We've got the air temperature in Celsius going from 0 to 22. We've got the metabolic rate, so that's how much oxygen it's consuming. It's not doing photosynthesis, it's doing cellular respiration. So it's consuming oxygen, and we can see that the enzymes, the part that would be increasing up to this point is missing because we don't have this metabolic rate until you get above freezing, and then from there on we see a linear decline between the air temperature and the rate of oxygen consumption. So the topics we're going to be talking about, we're going to give a little introduction with an example of Arctic tundra plant, the driest plant. Then we're going to talk about the impact of microclimate, which is extremely important in your temperature regulation. We'll talk about evolutionary trade-offs, then look at how temperature affects the performance of an organism, look at how temperature can be regulated within the body, and then how organisms have adapted to survive extreme temperatures. So that's the end of section 5.4, and there's some review questions at the end of that. Make sure you can answer the first two review questions. The one about the Celia, and the one about behavioral thermoregulation. Okay, so then we are moving on to how do organisms survive extreme temperatures? And there's a hint. We've got these beautiful monarch butterflies that obviously move across North America with the temperature. So there's a few ways. My favorite way to survive extreme temperatures is the simplest way, and that is to seek shelter. If it's too hot or if it's too cold, try to find some place where I can get out of the heat and the cold. Another way is to reduce your metabolic rate. Something like a hummingbird can, at night when it gets cold, can actually go into a state of torpor, which is where you have a very low metabolic rate and a low body temperature. And that way the hummingbird is going to save some energy when it goes into that state of torpor. Sometimes you can go into hibernation. So instead of just going into torpor for a few hours at night like the hummingbirds do, some go into a hibernation state for several months. And during hibernation, the body temperature can drop and the metabolic rates can drop. And that way the animals are going to save energy up during that period when the temperatures are not to their advantage. If we hibernate during the winter, if we go into this state in the winter, it's called hibernation, but some organisms will do it in the summertime. It's called estivation, and that might be something they're doing during the hot, dry periods of the summer. So the topics we're going to be talking about, we're going to give a little introduction with an example of Arctic tundra plant, the driest plant. Then we're going to talk about the impact of microclimate, which is extremely important in your temperature regulation. We'll talk about evolutionary trade-offs, then look at how temperature affects the performance of an organism, look at how temperature can be regulated within the body, and then how organisms have adapted to survive extreme temperatures. So there's a chapter. We talked about microclimates. We talked about aquatic temperatures, water moderating the climate. We talked about this important principle of allocation, that you're well adapted to certain environmental conditions, but not well adapted to all of them. The temperature and animal performance we talked about. We talked about extreme temperatures and photosynthesis. We talked about temperatures and microbes. We had the equation of balancing heat gain and heat loss, and we had a few examples there. How we regulate our body temperature in plants, ectotherms, endothermic animals, and some behavioral mechanisms for surviving extreme temperatures. So reread your chapter, take a look at the concept review questions in the sections, and then look at the chapter review questions at the end, 1, 2, 4, 6, and 8. So that's the even numbered ones. Make sure you can answer those. Also, take a look in Appendix A at Investigating the Evidence 5 about lab experiments, because we talked about quite a few lab experiments in this chapter, and be able to answer questions 1 and 2. Here's an example. Homework question. Butterflies are ectothermic and diurnal. If you don't know what the terms mean, look them up. What does it mean to be ectothermic? What does it mean to be diurnal? They elevate their body temperature by basking. If you don't know what basking is, look up. Look up words you don't know. How would the percent time spent flying versus basking vary with latitude? Could you draw a graph of that? Could you draw a graph with latitude being the independent factor and the percent time spent flying versus basking? And how does this happen? What if temperature changes over the course of the day? How much time would be spent flying versus basking? So you're changing the independent factor on this one as well. And then think about if this is the butterflies pollinating things, how is temperature then, since it's affecting flying versus basking, how is it going to affect pollination? The next chapter is going to be water relations. So I'm going to have you read the chapter. The concept review questions for the first and second sections of the chapter are good. The chapter review questions at the end, and then take a look at investigating the evidence in Appendix A, number 6, where you're looking at the sample size. So when you're reading through your chapters, you should be taking notes during the presentations, and you should write out answers to your questions. So that way you understand how to fully put the answers together. And see you next time. So the topics we're going to be talking about, we're going to give a little introduction with an example of an Arctic tundra plant, the driest plant. Then we're going to talk about the impact of microclimate, which is extremely important in your temperature regulation. We'll talk about evolutionary trade-offs, then look at how temperature affects the performance of an organism, look at how temperature can be regulated within the body, and then how organisms have adapted to survive extreme temperatures.